Time is more important now. Just go live.
Just check. I'm live.
Yes, you are. Welcome, Dr. Sandeep. Please start.
Thank you, Prof. Akshit. Thanks for asking me to talk about this topic. Essentially, I'm going to talk about viral inhibition by sodium chloride. And we've identified that we've identified a novel innate immune mechanism in our cells. So essentially, the cells are able to make use of the chloride ion from salt and have an antiviral effect. So I'm going to present the laboratory and the clinical evidence. I know you wanted me to talk about the clinical evidence, but I'd like to talk about the lab evidence first, because that'll make it'll help to understand what's going on inside the cell, and then we can come to the clinical evidence if I can. So the problem with most viral infection is we do not have a generic antiviral. Unlike a bacteriology, if any of you microbiology colleagues will know, you have gram-positive cover, you have gram-negative cover, but we don't have anything similar for viruses. And you have specific drugs for viruses, you have specific vaccines for viruses, but you don't have anything that covers viruses on a generic basis. For example, you can broadly classify viruses as DNA viruses, RNA viruses, enveloped viruses, and non-enveloped viruses. So there's nothing that you can give for DNA viruses or RNA viruses or enveloped or non-enveloped viruses. But why is that important? It's important because most viral infections are probably diagnosed by the general practitioner as a viral infection. Very, very rarely do you get a diagnosis as you're infected with metapneumovirus or RSV or coronavirus or HIV. So those are specific diagnoses. For the vast majority of infections, most of us come across upper respiratory tract infections, and we will all be most people will say you've got a viral infection. So without a generic antiviral, we are unlikely to be treating all these viral infections. So I'm just going to tell you a story. When I came to the UK in 2002, and in 2005, I was doing my MRC PATH exams, and just before my exams, I had 2 weeks of study holidays, and I was preparing for that. But bang in the middle of it, I had to go for an ENT procedure. So went there for a surgery, and post-surgery, they didn't give me any antibiotics, but they gave me a pot of sodium bicarbonate, and they gave me a sheet of instructions as to how to do nasal irrigation and gargling. So they told me to do that. As I came back and I was trying out nasal irrigation and gargling with sodium bicarbonate, but I didn't like the taste of sodium bicarbonate, and it didn't help me. In fact, the infection was bad. It was pretty severe. So I couldn't prepare for the exams. So in desperation, I thought, if I've got a bad sore throat, I would use salt water to gargle, because I've always gargled with salt water, very commonly done in India. And so I did swapped the sodium bicarbonate with sodium chloride, and I did nasal irrigation and gargling. And by the end of the day, I was feeling a whole lot better. So the next time I had a common cold, not only did I do the gargling, I also did the nasal irrigation part of it, because by then I knew how to do nasal irrigation. The rationale was this. Now that's the cross section of your head, and that's your mouth, and that's your nose. When you do gargling, this is the part of your throat you are treating. Whereas when you are doing nasal irrigation, that is the part of your nose that you're treating. And guess where most of the infection is when you have upper respiratory tract infection? It is usually there. So when you do nasal irrigation and gargling, you are in fact treating the entire upper respiratory tract. So since then, trial and error, I now know what works for me in terms of how many times to do nasal irrigation, what concentration of salt water to prepare, all that stuff. And most of my colds probably end up getting over in probably a day, maximum 2 days. And then I was interested in why it was working for me. And I came across a few papers, as you can see here. This is the picture of a person doing Jalaneti. very ancient yogic practice. It's there in an ancient textbook called Hatha Yoga Pradipika. If you check that out, you'll find something on Jalaneti. And there's a one-liner there which says, when you have a common cold, please do Jalaneti. And when you do Jalaneti, you don't just use tap water. You just don't use water. You always add salt to it. And I was surprised that Rabon et al. Taught woodworkers in Australia to do Jalaneti. And they did it, I think, from memory 3 times a day. And he went and asked them, are you guys happy to, at the end of the month, he asked them, are you guys happy? The vast majority said yes. And at the end of the, and he said, would you like to continue doing this procedure? And the vast majority said yes. The reason why Rabon was asking them to do Jalaneti was because they work in a very dusty environment and the wood particles irritates the nasopharynx. And so essentially, they were getting loads and loads of colds and lots of infections and other complications due to the dust particle. So when he followed them up 1 year, for 1 year, he was surprised to see that those who did the nasal irrigation had significantly fewer sore throats and colds. So that's one. We know patients, there's also another paper on children. I won't go into that for now. I came across this paper on cystic fibrosis by Elkins. Now, patients with cystic fibrosis, they have there's problems with the mucus, and it's pretty difficult to cough out. They get lots of infections, and they get nebulized hypertonic saline. When Elkins gave nebulized hypertonic saline was usually given as a mucolytic effect. It breaks down the mucus, so they're able to cough it out a lot quicker. So when Elkins gave nebulized hypertonic saline as a prophylactic measure twice or thrice a day, he found that those who had nebulized hypertonic saline had fewer exacerbations. And most exacerbations of cystic fibrosis are triggered by usually a viral infection. So the question I had was, is the salt water in some way preventing the infection, and therefore they're getting fewer exacerbations? Bronchiolitis, again, the use of nebulized hypertonic saline is being debated. There are groups in Israel and Australia who swear that children get better quicker if they get nebulized hypertonic saline, and there are other groups who swear that it does not work. So there's a bit of a debate going on, and they're collecting a bit more data on that. And then I was also looking up further information, and I came across articles from the 1960s. I'm just putting up one article here by Spear et al. And what they were doing was they were trying to make the virus stable by different means. They were using different pHs. They were trying out different sodium chloride, so the different salt concentrations. The idea was if you can add different salts or change the pH and make the virus stable, then vaccine can be stored at room temperature. You don't need a fridge for that. And Spear et al. Particularly was involved with a lot of work on Mengo virus. And Mengo virus is essentially it's an encephalomyositis virus, it's an enterovirus, very similar to the rhinovirus enterovirus family. And in this particular article that I'm showing you here, Spear tried out different sodium salts, different chloride salts, and different halide salts. And so he exposed the virus to the salt for 2 hours, and then he infected. It was, I think, it was a mouse model, and he infected the mouse. And when he was looking for a reduction in viral titers, as you can see, all the sodium salts stabilized the virus. There was no drop in viral titers. These are all in log values. But for sodium chloride, where there's a 4 log reduction in viral titers. All the chloride salts dropped viral titers by almost 4 logs. And amongst the halide salts, sodium chloride, sodium bromide, and sodium iodide dropped the viral titers. Sodium fluoride did not. So in 1961, Spear writes in his discussion, either there is a direct effect of the salt on the virus or there is an antiviral effect. So they stopped using sodium chloride to stabilize viruses. Clearly, it wasn't making the virus stable, but they didn't look at it from the antiviral side of things. And then I came across this paper from 2011 where they were investigating, they were essentially, it was a it was a paper on a particular dye for hypochlorous acid. And what they did was they took some bacteria, they ground it up, and then they added some sugar and fed that sugary solution to fruit flies. Fruit flies drank up the solution because there was sugar in it. But 1 hour later, they took the fruit fly, cut open the fruit fly, and stained the gut epithelium, which is what you see here, the last one. So this is the gut epithelium of the fruit fly, chock-a-block full of cells producing hypochlorous acid. So 1 hour after these cells were exposed to the bacterial lysate, they are busy producing hypochlorous acid. And hypochlorous acid is nothing but the active ingredient in bleach. So in lay terms, these cells, 1 hour after they were exposed to the bacterial lysate, are busy producing the active ingredient in bleach. And bleach, we know, kills everything. So which is where my hypothesis came in. When I do nasal irrigation and gargling, I'm supplying the cells with sodium chloride. Are the cells then taking up the chloride ion and producing hypochlorous acid? In which case, it has to work across the board, whichever virus we throw at it. So that was the hypothesis. So whether chloride salts could help suppress viral infection, whether it is mediated through increased hypochlorous acid production, and in which case could it be used against different viruses. So we tested that out. And I'm just going past this slide. Essentially, it just explains how broadly how we did these experiments. We went with HSV1. I went and met one of my colleagues, Professor Jürgen Haas, and in whose lab all this work was done. And luckily for me, he listened to my hypothesis, and we decided to go ahead with it. And we use HSV because Jürgen's lab uses HSV very commonly, and he had a particular type of HSV which had a green fluorescence protein. When the virus multiplies, the amount of fluorescence increases. So this is the first experiment that I'm showing you here. So what you see is, this virus was cultured over 48 hours, and this is the fluorescence of the virus. Okay, and so as you can see, this is HSV1 happily growing in the absence of any extra sodium chloride. So as the virus multiplies, the amount of fluorescence increases. And then we started adding 10, 20, 30, 40, 50 millimolar sodium chloride. And at 100 millimolar, I'm sorry, someone, and at, okay, 10, 20, 30, 40, 50 millimolar sodium chloride, you can see the drop in viral titers. And at 100 millimolar, the sodium chloride is completely, the virus is completely not multiplying any longer. So, but clearly you can see a dose-dependent reduction in viral replication here. So the question is it the salt affecting the virus directly, or because the viruses have to be grown in cells, could the salt be affecting the cells? So we checked for the viability of the cells, and we exposed the cells for different salt concentrations, 0 to 100 millimolar, and we exposed the cell for 24 hours and for 48 hours to sodium chloride, and we looked for the viability of the cells to see if the cell was actually viable. And as you can see, anything above this threshold, 70%, is viable. Even at 100 millimolar sodium chloride, the cells were fine. So it means the salt is not necessarily affecting the cells, but is in some way preventing the virus from replicating. So the question was, how exactly is the virus being killed? So is the salt affecting the virus directly? Or is the salt preventing the virus from binding to the cell? Or is something happening within the cell? So we decided to test all these three things, and we exposed the virus to sodium chloride for 0, 1, and 2 hours, and then we added the virus to the cell. Okay, so we exposed the virus for different salt concentrations, 0 to 100 millimolar. And to give you, I'm just going to show you this one, 100 millimolar. The virus was not exposed to salt. They exposed to salt for 1 hour, exposed to salt for 2 hours, and then the virus was grown. As you can see, whether it was exposed for 0 hours or 1 hour or 2 hours, there is no reduction in viral titers. Okay, meaning it is not a direct effect of the salt on the virus. The next question is, does the salt prevent the virus from binding to the cell? So we added the salt just when the cell or when the virus was binding to the cell. So which is what you see here, the orange bars here. With increasing salt concentration, all the orange bars are equally high, meaning there is no reduction in viral replication when the virus was binding to the cell. And then the last question, what is happening? Is it something that is happening within the cell? So we added the salt only when after the virus is bound to the cell and has got inside the cell and is now multiplying within the cell, we added the sodium chloride. And that's what you see with the blue bars. With increasing salt concentrations, you can see the blue bars, it is coming down, the viral titers go down. The gray bars was when we added the salt both during adsorption when the virus was binding to the cell and during replication when the virus is multiplying within the cell. It is very similar to the blue bar, in fact slightly better. So it is not, the salt is not killing the virus directly. It does not prevent the virus from binding to the cell, but something is happening inside the cell. So the next question is, when you add sodium chloride to water, sodium chloride breaks up into sodium ion and chloride ion. And then sodium ion goes into the cell through the sodium channels and chloride ion goes into the cell through chloride channels. And we thought, let's block the channels and see what happens. So this is the virus happily multiplying in the absence of any extra sodium chloride. And then we added 50 millimolar sodium chloride and the viral titer goes down. And then you start blocking the chloride channels with NPPB. So that's the chloride channel blocker. And you start adding increasing concentration of NPPB and slowly all the chloride channels are being blocked. When all the chloride channels are blocked, the virus that was originally inhibited comes back. Meaning if you prevent the entry of chloride ion into the cell, the virus is happily able to multiply. We block the entry of sodium ions using sodium channel inhibitors. We used alphanamide and benzyl amiloride, and in both cases the virus does not come back, meaning it is not the sodium ion that is relevant, it is the chloride ion that is relevant. The question is, what is happening to the chloride ion? The hypothesis was the chloride ion gets converted to hypochlorous acid. So we decided to measure the production of hypochlorous acid, and which is what you see here. With increasing sodium chloride concentration, there is an increase in amount of hypochlorous acid production. We've tested that with 2 different dyes. Both are specific for hypochlorous acid, and both show an increase in hypochlorous acid production. So lastly, now chloride ion has to get converted to hypochlorous acid through a peroxidase enzyme. So we thought, let's block that enzyme. So this is the virus happily multiplying. We add sodium chloride, the viral titer goes down. And then you block the peroxidase enzyme with increasing concentrations of 4-aminobenzoic acid hydrazide. And what happens is the virus comes back. So even if you stop the conversion of chloride to hypochlorous acid, the virus starts multiplying again, meaning it is the hypochlorous acid is the active ingredient that is able to inhibit the virus. And all we're doing is, if we supply the cells with the chloride ion it needs, it is able to produce a hypochlorous acid and therefore inhibit the virus. So if that is the case, it has to work whichever virus we throw at it, because hypochlorous acid, like I told you, is the active ingredient in bleach, and bleach we know kills everything. So we tested that against HSV. Remember the first experiment I showed you, a clear reduction in viral replication. VZV, similar story. RSV, influenza A. Now HSV and VZV are DNA viruses. RSV is a RNA virus. Influenza A, RNA virus. All 4 are enveloped viruses. Coxsackie virus B3, similar story. This is an RNA non-enveloped virus. And coronavirus 229E, it's a coronavirus, RNA and enveloped virus. So whether it's a DNA virus, RNA virus, enveloped, non-enveloped virus, it works across the board. So in summary, sodium chloride has a dose-dependent antiviral effect, and the effect happens within the cell. It needs the chloride ion, and we've shown that the chloride ion has to be converted to hypochlorous acid. And if you block the conversion of chloride ion to hypochlorous acid, the virus is happily multiplying. Therefore, chloride salts can be a therapeutic antiviral agent. So we published this paper in 2018 in Scientific Reports. And whilst all this was going on, we conducted a pilot study of hypertonic saline nasal irrigation and gargling to see whether it actually works in clinical practice. And the study was called the ELVIS study because it has to be something catchy. Initially, I thought hypertonic saline nasal irrigation gargling, HISNIG study, but that was not very popular. So we decided to go with this name, Edinburgh Lothian Viral Intervention Study, which stands for ELVIS. People remember Elvis Presley. So we went with that name. And the aim was very simple. I know I gargle, I know I do nasal irrigation, but I wasn't sure whether people in Edinburgh will be happy to do that. So essentially, we wanted to see whether we could recruit people for the study. And then the secondary outcome measures included duration of symptoms and viral shedding. And we decided we will need at least 30 per arm to analyze the results. So we decided to recruit a maximum of 80% so that we have feedback from at least 60 individuals. So I'm not going to take you through the sample size calculation and stuff. Inclusion criteria, everybody, any adult who had a cold, as long as the cold was within 48 hours of onset of the cold, they could join the study. We just told them they had to be squeaky clean, i.e. healthy adults. They shouldn't be pregnant. They shouldn't have any chronic or immunosuppressive conditions. They shouldn't have allergic rhinitis. So essentially, just to make sure that who the people we are dealing with are people with a common cold. And they had to keep a diary for up to 14 days or until they were well for 2 days. So they kept this daily diary, which is this is a WRSS 24 diary, Wisconsin Upper Respiratory Symptom Survey diary, and we added a few other questions to that. And the intervention arm was taught how to make salt water, ideally to use 3%. That is 3 grams in 100 ml of water. Or if they found that too salty, then they could reduce the concentration a bit. And the videos are here in elvisstudy.com. We taught them how to do a nasal irrigation and gargling. Again, the video is in elvisstudy.com. We just gave them a sea salt. We gave them measuring spoon, bowls, and a flask. All that we told them was to use the nasal irrigation and gargling as many times as needed. And the only issue was they had to boil the water first, add the salt, wait for it to cool, and then they are good to go. They can either make it fresh every time, or they can make it in bulk in a flask, and then they can use that for 1 day. And but they have to keep using fresh water every day. So the videos are there in elvisstudy.com. It'll be there for another couple of days, but we'll probably have to remove that because we are starting another study now. So both the groups were also asked to collect nose swabs on 5 consecutive days. So they essentially take a swab from the nose on 5 days, first thing in the morning, and they put it in this box here, and they post it back to the lab for testing, because we can then measure the amount of virus that is in the swab. So as you can see, we recruited 30 per arm, intervention arm and control arm. And if you look at the baseline characteristics, is very similar between the both arms. There was no major difference there. And the vast majority of the intervention arm were happy to use 3% hypertonic saline. Some found that a bit too salty, so they went for 2.5% and 2%, just about 9% each. And now, this is the symptom over 14 days, how unwell they were. And it was graded from 0 to 7. Okay. And this table here is the control arm, and this is the intervention arm. Now, each line represents the symptom score of one person that day. Okay. Even visually, there's a huge difference between the intervention arm and the control arm. And if you look at the duration of symptoms, there's a 2-day reduction in duration of symptoms in the intervention arm compared to the control arm. If you look at individual symptoms, runny nose, blocked nose, sore throat, sneezing, scratchy throat, and cough. And as you can see, the previous slide, this difference in 2-day reduction in duration of symptoms was statistically significant, despite the fact it's a pilot RCT. Runny nose, sneezing, blocked nose, and cough were also significantly lower in the intervention arm. Some were not significant, even though it looks significant, it looks different, but statistically it wasn't significantly different. Sore throat and scratchy throat. Hoarseness of voice was again significantly lower in the intervention arm. Feeling tired, head congestion, chest congestion were also measured. And this shows you how many times the person did nasal irrigation. We had told them to do it as many times as needed. So the rationale was you do the procedure, you feel better for a couple of hours, and then the symptoms come back. That's when you repeat the procedure. So the first day, you would expect them to do it a lot more times. And as they felt better, they did it fewer times. That is very similar. This picture is very similar to the question how unwell they felt. Very, very similar pictures there. So in total, they did it for a median of 5 days and a median of 3 times per day. Then we also got feedback from participants. We asked them if they took a medication for the cold, significantly lower in the intervention arm compared to the control arm. And we asked them if there were anybody with symptoms after them, i.e. transmission. So if there was somebody in the house who developed a cold after the index case, significantly lower in the intervention arm compared to the control arm. So did it make a difference? Significant. 93% said yes. Will you use the procedure? 60% said yes. 25% they were not sure. And if the procedure were more convenient, all the 25% said likely. Okay. And procedure is comfortable or moderately comfortable. Then we measured the virus. As you can see, the vast majority of people were infected with rhinoviruses, which is one of the commonest causes of the common cold. But the second commonest ones were coronaviruses. Okay, there are 4 other coronaviruses apart from the one that is circulating right now. These 4 coronaviruses are very common across the world. Okay, and they are the second commonest cause of upper respiratory tract infection. In fact, we also had people with flu, RSV, parainfluenza causing the common cold, because the common cold is a clinical entity, not a virological entity. So a large number of viruses can cause symptoms of the common cold. So if you look at, so this is the viral shedding data. Now this side is from the control arm, and this side is from the intervention arm. The gray area is how unwell the person felt. The orange line is the viral shedding. And for each person, this is for each person. For each person, I've given which virus the person was infected with. Okay. And on the intervention side, you've also got these columns there, which represents the number of times the person has done nasal irrigation. So the viral shedding, the orange line, that value is actually log value. So it looks small, but it's actually each 1 point is one log reduction. Now, as you can see, this gentleman, this person here, there was a drop in viral shedding, and that mirrors the number of times a person has done nasal irrigation, and also the symptom score. Same there, same there, same there. If you look at these individuals here, this person has done nasal irrigation on 3 days. Viral load is coming down. Like I told you, this is log value. So this is actually quite a good fall in viral load. And then the person stops doing nasal irrigation, viral load goes up again. Same story here, same story here and here. And this person has almost cleared the virus, stops doing the procedure, viral load comes back up and starts doing the procedure again. So we then measured a fall in viral shedding, and the intervention arm had a higher rate of fall compared to the control arm. And that was again statistically significant. So in conclusion, clearly it's a pilot RCT. We needed a larger study with the efficacy endpoints, but yes, we are able to recruit people, and is it acceptable? And there was a significant reduction in duration of symptoms. We published this paper as well last year. Now, after COVID-19 happened, we went back and reanalyzed the data, looking at just the coronavirus cases in our study. Now, that is your intervention arm, that's your control arm. Very small numbers, just eight, I think from memory, 8 people here and 7 people here. And you can see it looks different. There was a 2.6 day reduction in duration of symptoms in the intervention arm compared to the control arm, but it was not statistically significant. P is 0.054. It is just beyond the 0.05 threshold. So, looking at the coronaviruses, we had a collection of both alpha and beta coronaviruses, and so essentially it works in both these types of coronaviruses. So which we published, we analyzed the data and we published this data as well. So which is why we've said it should be considered as a treatment option for COVID-19. So if you, having listened to all this talk, I would like you to go back and read up this paper as well, A Pinch of Salt by Kirsty Minton. Now, Kirsty's reviewed an article by Jansch et al. And Jansch has shown that epithelial cells accumulate sodium chloride. And if there is an infected arm and an uninfected arm, the infected arm accumulates a lot more sodium chloride than the uninfected arm. And through what they've shown is, interestingly, through the sodium route, there is an antibacterial and an antiparasitic effect. Okay. And they've also shown that in a animal model, they've introduced an infection, and they've, I think from memory, it's from leishmaniasis, and they applied sodium chloride, and there was the infection got better. I just need to clarify that infection because I'm talking from memory here. So what they've shown is through the sodium route, there is an antibacterial and an antiviral effect. But what we have shown is through the chloride route, there is an antiviral effect. So sodium chloride, therefore, possibly has both an antibacterial and an antiviral effect. So the possibilities from this data is essentially we need a larger study in adults. We are now doing a larger study in children. It's called the ELVIS Kids study. We were halfway through the study. We've recruited 230 odd children so far. We need to recruit another 250 more kids. But after COVID happened, we had to stop the study. So we will start again when we're given the permission to start again. We are now awaiting approval to start a COVID-19 study in people who are self-isolating. So it's gone for ethics. So we are waiting for feedback from them. We can try it out in different other scenarios. For example, one of the things I will be interested in testing is whether diarrhea. You can use, for example, lassi with salt. Most of us, our grandparents would have given us that. Buttermilk, curd and rice with rice and curd with salt when people have an upset tummy. So there are loads of ways we can try these out. Loads of cold sores, for example, very, very useful. The first virus that I showed you, HSV, actually causes the cold. So quite a few implications for this data and this research. This is the ELVIS Kids study that we are halfway through, and hopefully we will complete the study next year. And before I complete, I'd like to thank quite a few people, particularly Jürgen, who actually, whose all the lab work was done in Jürgen's lab, and for supporting me through all these years. And to Aziz and Kat and Lynn, who are my colleagues who were involved with the ELVIS study and who are also involved with the ELVIS Kids study. And to Copan, who gave us swabs, and Cornish Sea Salt, who gave us the sea salt for free. That's me. Thank you so much.
Thank you. Can you come online from your slides so we can see you?
Yeah.
Thank you very much for a beautiful talk. Nice, comprehensive. Before I go to a couple of people who are listening to you, we cannot go to all the Facebook viewers, but we do have certain people from our lab, from Harvard Medical School, Dr. Sadbir. I can see him around. Before I asked them, I was just wondering, why didn't you call it ELVIS JN study, a Jalneti study?
No, the reason why I didn't call it Jal JN study was mainly because it wasn't Jalneti. Jalneti is when you're pouring the water through the neti pot. So we're just giving them a bowl, and they're just sniffing it up. So that's why it's called ELVIS COVID-19.
Right.
And also the WHO wanted everything to be have that name COVID-19 associated with the study.
Sure.
Yeah.
So I'll open it to the panelists, to the students, to Dr. Sadbir. If you have any questions, students have any questions to ask. That was a very good study from going on from 2019. Now they're revisiting the data in respect of COVID-19. Do you have any questions about the cell culture experiments or any of those graphs that he showed you? Dr. Sadbir, do you want to ask?
Yes, absolutely. Fantastic presentation. And I'm very impressed with the in vitro work, which worthy of publishing in a Nature journal, how you very elegantly nailed down the intracellular chloride role. So that's fantastic. That's such a very strong support. The other comment is that in my literature searching on this topic is that nasal irrigation based upon Jalanetti has been known in Western medicine for over 100 years. In fact, there's a publication in Lancet in the late 1800s talking about the different methods of nasal irrigation. But what's surprising is that this result on the effects of upper respiratory tract infections has not been known until that first study in 1999 with the woodworkers. And so I wanted to just ask to reflect on your part. You were given sodium bicarbonate instead of sodium chloride, and you switched to sodium chloride. Maybe you can reflect on the significance of that and the loss of efficacy that was there in these nasal surgeons in prescribing sodium bicarbonate instead of sodium chloride.
I mean, they were essentially, I presume they are using sodium bicarbonate mainly to make the solution isotonic. Because tap water will be hypotonic. So if you sniff hypotonic solution, it'll hurt. So they were giving us some salt to make it less stingy. But I hated the taste of sodium bicarbonate. That's one. And I also had an infection which didn't go away. So which is why it was just by chance that I thought if I have a bad sore throat, I use sodium chloride. So why don't I swap? So which is what I did. So it was by chance.
I want to come back, yeah. I want to come back to your slide. If you remember, you showed that it does not kill the virus. And then you showed the couple of experiments that it does not prevent the entry of the virus either. So for the simplification, I know at the end of the talk, you mentioned that there is an accumulation of more and more of sodium ions. Or in one case,
that is Jan Chattal's paper.
So in terms of
on epithelial cells.
On epithelial cells. So when you showed that there is this does not kill the viruses and it doesn't prevent the entry of the viruses, for the purpose of simplification, because we have a interdisciplinary gathering here, people from yoga field, from some are physicians, some of them are scientists, some of them are students. So can you explain this? Can you elaborate this in a bit of more detail? How exactly does it work?
Okay, I mean, for I was explaining the accumulation was a different thing. That's for what Jansch has shown, that our cells apparently selectively accumulate sodium chloride, and they've measured the sodium ion, and they've shown that through the sodium route, there is an antibacterial effect. So that's a different thing altogether. What we have shown is we have taken the virus and we added salt to it, and we kept the virus in the salt mixture for 1 hour or 2 hours, and then we infected the cell. If the salt is able to kill the virus, when we infect the cell, the virus should not be able to multiply. Okay.
Yeah.
Whereas, so when we do it that way, the virus is happily multiplying. There is no change. And we've tested that at different salt concentration. So it is not that the salt is killing the virus directly. Next, when we grow the virus, we always, assuming this is a layer of cell, we apply this, we put the virus on the cell as a solution. Okay, so then the virus sits on the cell, and then it has to bind to the cell, and then it has to get into the cell. So what we did, we added the salt, added the virus, and then we added the salt. So the virus was sitting along with salt to see whether it can prevent the virus from binding to the cell. But that did not prevent the virus from binding to the cell. Next, we only added then we added this virus to the cell. It got into the cell and is multiplying inside the cell. And then we topped up the salt. Then the salt goes into the cell, and then then the antiviral effect starts happening. So that's how we show that it is not a direct effect of the salt on the virus. It does not prevent the binding to the cell, but something happens within the cell. And then because the salt is sodium chloride, sodium goes through the sodium channels. So if this is the cell wall, the cell membrane, you have pores through which the chloride ions and sodium ions go in. So if you can block those pores with a channel blocker, it can't go in, which is what we did. So we prevented the entry of sodium ion and chloride ion. Then you realize when you block all the chloride ion, the virus comes back. And then we also showed that once the chloride goes in, it gets converted to hypochlorous acid, which is the active ingredient.
Great. Are there questions from any of the students here? Dr. Sadhvi, do you want to ask him if there was difference in the outcome between people who did NETI once, twice, or thrice? Dr. Sandeep, did you see any significant results? And number two, did you do the same experiments COVID-19? Probably not yet.
We have applied for ethics for that. And it's come back with a few questions. We've just responded to them. So hopefully next week we should get approval. We're just waiting for that. Once the approval comes through, then we'll be starting that study in COVID-19.
No difference between the frequency of doing this once, twice in a day, thrice in a day?
No, we haven't done it that way because in our study, what we told them to do was to do it as many times as needed. Because personally, when I do the nasal irrigation, I find it, I get better quicker. I feel better for another 1 to 2 hours, depending on how bad the cold is. And then I feel the symptoms coming back. And that's the time you repeat it. So essentially, when you do nasal irrigation, at the end of the procedure, the cells are left with a very thin layer of salt water. And then the chloride ion goes in, and there is an antiviral effect. So my hypothesis is you feel better when there is an antiviral effect going on. And then there is not enough chloride ion, the virus starts multiplying again, which is when you start feeling those symptoms coming back. At that time, you go back and reapply the salt water. So the number of times you do it depends on how bad your cold is. So we didn't tell them to do it any number of times. We told them up to a maximum of 12 times, do it as many times as needed, which is why in the graph I showed you one graph where they did it up to 8 times on the first day, and as they felt better, they did it fewer times.
Yeah.
Yeah,
So
there was a.
Please go ahead.
Also, looking at the literature, there was a Swedish study, and instead of using a nasal wash, they used a sodium chloride nasal spray. And in that study, they also found reduction in the amount of upper respiratory tract infections.
Is that the one in children?
And then there's one in children by Slepak and colleagues.
Yeah, Slepak,
and that's the one that showed reduction in illness and absence from school and so on.
Yeah.
So that was a more direct, that was a nasal wash.
Yeah.
And I think your study is probably replicating that to a large degree.
Yeah. But thing is, with both the Australian Rabon study and Slepak study, they did actually measure the virus, which is what we've shown here. So
exactly.
That's the one thing.
Yeah, they had indirect measures of the duration and severity of the illness. But yeah, absolutely, looking at viral measures is very important.
Yeah. You mentioned something about a paper from the Lancet in the 1800s. Is it possible to send the reference to me?
I happily do that. Yes.
Fantastic. Yeah. Brilliant.
I have one more question from the Facebook. This is not among the panelists or the students. I was just looking at my iPhone. Dr. Kaushal Sharma has a question for you. Okay. Does NaCl exposure has any role on the adaptive immune response pertaining to viral neutralization or cytotoxic activity of TC cells?
I would doubt that, because essentially it is an innate immune response in the so this is the innate immune response in each and every cell that is lining your upper respiratory tract, right? So in a way, yes, it will have an adaptive immune response because the larger the amount of virus that is circulating in you, the stronger the response that you need to get rid of it. So yes, in a way, it will, you'll probably produce a lot more antibodies and a lot more T cells and stuff if you have a much more serious infection. But just similar to a vaccine, where if you give an attenuated virus, you do mount an immune response. The person is actually infected with the virus, a wild type virus in this case, and you're just suppressing the infection a little bit, but they will definitely be mounting those immune responses, adaptive immune responses. Yes, both the antibody and the T cell responses will be there. So when they come across the infection the next time, they will have that. They will have the benefit of that. But you're just bringing down the severity of the infection the first time around.
I have one more question, and then we'll let you go. And the question is a little bit different from our subject of discussion. It's more about the lab's environment. You know, there are animal studies going on, and then there are cell culture studies going on, and there are people now. We have started working again in the lab with social distancing. I was wondering, we do a lot of the neuronal cell cultures. In particular, motor neuron cell line is what we are culturing. So do you think the students or the staff which work on these cells, they do the cell cultures, when they go to the hospital, and if they are potential carriers, do you think that this may infect the cell cultures or the animals they work with? Should they use PPEs to ensure that there is no purification or enrichment of cell viral cultures in the cells they infect? Because we've been talking a lot of safety measures in our, the clinical colleagues talking about when they do the COVID duties, the protective gear and all that. So in terms of scientific lab, because they are not so away from the COVID block, they may carry some infection with them and carry it all the way to the cell culture.
That will be on par for any viral infection that they have. It could be a rhinovirus, it could be a metapneumovirus, any of those infections. You will have the same thing. And I don't think it is that simple that you'll be able to just breathe and the virus grows into the cell culture. Because even when, if that's the case, all our cell cultures have to be contaminated. That's not the case. And it also possibly depends on the type of cell that you use. Some cells are a lot more resistant to others. I'll be very surprised if just because someone is asymptomatically shedding, the cell cultures will get contaminated with that virus. Because if the virus is there in the air, you need to purify the virus. You have those, to sample the air, you have those huge machines which purify the virus, and then you culture it, because it's going to be in very, very small amounts. It's unlikely to be a major
No, but you
issue.
can carry it to your hand on the surfaces. And if you contaminate your gloves, and you'd start seeding the cells and splitting the cells. And
if the person who's doing the lab work is not paying attention to that, then I think much more than the viral contamination, you'll probably have a lot more bacterial and the fungal contamination in the cell cultures,
sure,
a lot more commonly than the viral. Because even before the virus grows, I'm sure the bacteria will grow, and you will not have a normal cell culture to go with. Mycoplasma is a very common issue in cultures. So that probably is much more an issue. And I'm not sure how well it grows in neuronal cells anyway.
I haven't got any literature. We tried to search, we didn't find any literature.
No, because it doesn't grow very well. I mean, we've cultured the virus in CACO2 cells, and it seems to grow very well. But in other cell lines, there are other cell lines where they haven't shown, they've shown that it doesn't grow as effectively. So I would be very surprised if someone asymptomatically shedding is able to contaminate the virus. I don't think that's been shown for any other virus. I might be wrong, but I don't remember seeing that.
Okay. So any more questions from any of our friends? Let me also quickly look at Facebook questions. Well, if there are none, then we must thank you for a wonderful presentation. Thank you for coming live all the way. And through
Thank
you,
you so
we
much.
were able to discover the beauty of this panel discussion being exported to Facebook. And I hope a lot of people pick up their containers and start doing, if not gel,
The only thing I would request is please boil the water. Please don't use plain
course.
water. Always boil the water first. Thank you
Yeah,
so
it
much.
is one of the Kriyas. Kapalbhati is one of them. Then there's Sutra Neti, Jal Neti, and people often do this and then follow it up by Kapalbhati and Pranayam or other asanas. So this is like a prelude to the real asanas and meditation.
I remember one of my classmates' father when I was a kid, when I was growing up I must have been about 10, 12 years of age. I remember my friend's father sitting outside the house and doing Sutra Netri. So I remember that.
We had three very nice practical sessions on this same platform by Dr. Sawant. He demonstrated very nicely how to do Jal Neti, Sutra Neti. Maybe you should consider looking at our archives sometime.
I should, yeah.
We'll
Thank you so
reconnect
much.
again. Thank you very much from our journal, Integrative Medicine Case Reports, and I request that you consider sharing at least some part of the data for our journal, which is focused on bringing the traditional and the modern science together.
Fantastic. Thank you so much, Prof. Akshay. Thank you.
Thanks.
Thank you. Thank you, Dr.
Thank you, Dr. Satya.
Satya Singh. Thank you.